Welcome to Biotech Academy, Class 2. Topic is Lipid bilayer and membrane protein diffusion. Proteins are embedded in the phospholipids. Bilayer have number of functions. They may include moving materials through the membrane and receiving chemical signals from the cell's extracellular environment. Carbohydrates are associated with the membranes, which are attached to the lipids or to the protein molecules. Carbohydrates may be covalently bonded to lipids or proteins. These are glycolipids and glycoproteins. Glycolipids are consist of a carbohydrate covalently bound to a lipid. They serve as recognition signals for interaction between cells. Glycoproteins. These consist of a carbohydrate covalently bound to a protein molecule. The bound carbohydrates are oligosaccharide chains. The main function of the glycoproteins enable a cell to recognize by other cells and proteins. Both glycoproteins and glycolipids are essentially abundant in the plasma membrane of eukaryotic cells. They are absent in mitochondrial membrane, chloroplast, lamellae, and several other intracellular membranes. The membranes are responsible for cell recognition and addition of huge membrane glycoproteins. These glycoproteins are 80% sugars. Human ABO blood group antigens are oligosaccharides. They are covalently attached to the glycolipids or glycoproteins in the plasma membrane. Anchor proteins are succinate dehydrogenase. Electrostatic binding to bilayers are myelin basic proteins. Hydrophobic interaction with bilayer E. coli pyruvate oxidase, anchor protein by cytochrome B5, signal transmembrane segments by glycoporins, multiple transmembrane segments by lactose permease ion channels, anchor proteins by eukaryotic alkaline phosphatases. These are some of the modes of membrane protein binding to the membrane. The lipids, which are consist of bulk of membrane. So most of the lipids in biological membrane are phospholipids. The phospholipids, some of the compounds are hydrophilic, these are water loving, and other are hydrophobic, that is water hating. Phospholipids have both hydrophilic region and hydrophobic region. Thus, phospholipids are called as amphipathic nature. Amphipathic nature. Hydrophilic region, which is phosphorus containing head and which is electrically charged and are associated with the polar water molecule. Hydrophobic region is the long nonpolar fatty acid tail of phospholipid which are associated with other nonpolar materials but they do not dissolve in water or may associated with hydrophilic substances. There are three classes of lipids that are found in biomembranes. There are most abundant phosphoglycerides, sphingolipids, steroids. All three classes of lipids are amphipathic molecules, have a polar hydrophilic head group and a hydrophobic tail group. The phosphologens are a group of phosphoglycerides. These are a group of phosphoglycerides which consist of 20% of the total phosphoglyceride content in human. These are abundant among tissues, species, in high human brain cells and head tissues. The complex glycosphingolipids are called as gangliosides. Complex glycosphingolipids are called as gangliosides. These are one or two branched sugar chains which consist of sialic acid groups which are attached to the sphingosine. So these are attached to the sphingosine molecules. They are most abundant in nerve tissues. Nerve tissues are made up of with gangliosides, that is glycosphingolipids. The third class is cholesterol. Cholesterol is especially abundant in plasma membranes of mammalian cells, but these are absent in most prokaryotic cells. So all mammalian cells which consist of 30 to 50% of the lipids, and come to the plant plasma membrane which consists of 30 to 50% of lipids, contains steroid unique in plants. So lipid bilayer has important properties. There are first one is hydrophobic, the second one is hydrophilic nature. Hydrophobic core is an impermeable barrier which is prevent diffusion of water molecules across the membranes. The second property is the bilayer is stability. So lipid bilayer has two important properties. The first is the hydrophobic core and the second one is the bilayer is its stability. The bilayer structure is maintained by the hydrophobic van der Waals interactions between the lipid chains. So van der Waals interactions between the lipid chains. Okay, the lipid bilayer has two important properties. The first property is the hydrophobic core and the second property is the bilayer has its stability. Bilayer has its stability by the hydrophobic maintained by the van der Waals interactions between the lipid chains. The next topic is lateral movement of proteins and lipids. You know that cell is consist of membrane proteins. Cell has membrane proteins, all these membrane proteins, right? The cell has lateral movement. The lipid bilayer, one lipid bilayer is there. So this lipid bilayer, lateral is nothing but the sides, the movement by the sides. The movement of the materials by the sides. One lipid to another lipid, another lipid to another lipid. Like that, the movement by the sides, lateral, that means side movement. Flip flop movement. Flip flop movement is nothing but the movement in rotation. Flip or flop movement, up or down. Okay, the next term, the fluid content of one lipid molecule may be that tails are unshaped. There is no proper shape of the tails in unsaturated hydrocarbon tail. So fluids contain unsaturated hydrocarbon tails. Viscous materials contain viscous membranes contain saturated hydrocarbon tails. Saturated hydrocarbon has straight tails. There is no shapeless. They are shape tails. Okay, the viscous is nothing but a little jelly structure of the membranes that contain saturated hydrocarbon tails. So unsaturated is fluids are contain unsaturated, viscous are contain saturated hydrocarbon tails. The next one is membrane protein asymmetry. Asymmetry is nothing but shapeless. Proteins are asymmetrically distributed on inner and outer surface of a membrane. They are asymmetrically distributed on inner and outer surface of a membrane. Integral membrane proteins that protrude on both sides of the membrane, hence known as transmembrane proteins. So they different faces on two membrane surfaces. So lipid which has different shape on two membrane head faces. The membrane faces are different shapes. That's why asymmetric shape. The proteins are asymmetric shape in membranes. The plasma membranes has one protein molecule for every 25 phospholipid molecules. This ratio varies depends on the membrane function. The inner membranes of the mitochondrion, which is specialized for energy processing, there is one protein for every 15 lipids. On the other hand, we can see that myelin, a membrane which is enclosed by the nerve cells and uses the properties of the lipids to act electrical insulator. There we can see that one protein per 70 lipids, like phospholipids. So membrane proteins has hydrophilic regions, hydrophobic regions. Hydrophilic regions which are stretched of amino acids with hydrophilic R groups, which give certain regions of proteins a polar character. Hydrophobic region stretches of amino acids with hydrophobic R groups with other regions of the proteins non-polar character. Hydrophilic regions has a polar character, hydrophobic has a non-polar character. The cytoskeleton have components just inner surface membrane and which are attached to the proteins. They protrude inside to the cytoplasm. Lipid rafts, groups of lipids are the state for the trap of proteins within the regions. Come to the functions. Functions of plasma membrane. The main functions are transport. The transport by the channel proteins. Channel proteins are the forms of channels for lipid insoluble molecules and ions to pass freely through membranes. Carrier proteins, which binds to a substance and carries across the membrane. Transport by channel proteins and carrier proteins. The next function is intercellular junction proteins. Intercellular junction proteins, these are proteins bind cells together. Tight junctions, desmosomes are the some of the protein bind cells together. Tight junctions are transmembrane proteins of opposite cells attached to tight zipper like fashion. There is no leakage between them. For example, intestine, kidneys, epithelium of cells, epithelium of skin. Next, desmosomes. Desmosomes are cytoplasmic plaques of two cells which are bind with the aid of intermediate filaments of keratin. The next one is signal transduction. Signal transduction bind to chemical messengers which are sending the messages into cells which cause the cellular reactions. Next one is catalysis. Catalysis carry out enzymatic reactions. Cell recognition proteins. Cell recognition proteins by glycoproteins or glycolipids on extracellular surface and serves as the cell recognition tags. Carbohydrates are short branched chains of less than 15 sugars. Addition. Addition is nothing but attached to cytoskeleton or the extracellular matrix. Extracellular matrix protein fibers and carbohydrates secretes by cells and filling the space between the cells and supporting cells in the tissues. The next one is gap junctions. Addition by gap junctions and connections. Gap junctions are channel proteins of opposite cells. They join together providing channels for ions, sugars and amino acids and other smaller molecules to pass. This allows the communication between the cells. For example, heart, muscle, animal, embryo. Connexins are the junctions which made up of with specialized protein channels. Each gap junction which is made up of with specialized protein channels is called as connections. The span of these plasma membranes of two adjacent cells in the intercellular space between them. They are involved in the dissolved molecules, electrical signals, and can be passed from one cell through other cells. These junctions are function in passing of the materials, electric signals, dissolved molecules. So additions are gap junctions and connections. Gap junctions are channel proteins of opposite cells joined together providing the channels for ions, sugars, amino acids and other smaller molecules to pass. Connections are specialized protein channels in gap junctions. Okay, these are about membrane proteins asymmetry. Thank you for watching. We can meet on next class. Keep write down this all content in your book, and when you're getting free time, you refer this all content, store in your brain. It will help you to crack CSER exam and other national exams. Thank you all. Bye. Signing up. Meena.